Genes, ecology, behavior. All three factors combine in the evolution of species. But what is random and what is directed, governed by natural selection? Because geneticists are unsure of the interplay of these two ideas, there's a long-running debate among biologists about how species originate and how they evolve into the diversity that we see around us today. These gulls, living on the island of Stockholm off the coast of Wales, have diversified into a second and related species. It's thought to be the result of selection, where geography has played a crucial role. The story behind the evolution of these two species lies in their present-day distribution in Europe and North America. Both birds descended from the same gulls that lived in Russia thousands of years ago. Some moved westwards into northern Canada and then crossed the Atlantic into Britain. Others moved first east and then west across Europe. Today, the two ranges meet again in Wales, reuniting the modern relatives. Although they're closely related, they no longer mate with each other. And they've become these two separate species, the herring gull and the lesser black-backed gull. Within the ring of their distribution, the gulls only gradually change in form, but at the two ends of the ring, they form perfectly distinct species. The two species look different. This is the herring gull, and this the black-backed gull, but they're distinguished by their isolating mechanisms that prevent interbreeding and lead to new species. Close study of the behavior of these species of gull shows that they rarely interbreed. So what is the isolating mechanism? To find out, researchers switch eggs between the two species. Will the parent of one species foster the chick of the other? Or are the species so different that they will not even bring up each other's young? It turns out that they will foster gulls of the other species. The foster children will go on to interbreed with their own foster relations, so there's no physical reason why they should coexist as separate species. It's a behavioral, not a genetic difference, which leads to mate choice within their own species. For the gulls, mate choice has led, through behavior, to the formation of a new species. But the way that species evolve can also depend on the size of the population. The population size of the cheetah is very small, and this has often been the starting point for new species. New species evolve following an isolation of one kind or another, and the rate of evolution is rapid. A lot is known about the numbers and distribution of the cheetah population. At the turn of the century in Africa, cheetahs were widespread, but today the picture looks very different. Why has the cheetah become so scarce? At the De Wild Reserve in southern Africa, breeding studies gave the first clue. The sperm of the male cheetahs in the colony were often deformed, sometimes with smaller heads, sometimes with kinked or distorted tails. But these were symptoms of a deeper genetic problem for the whole cheetah species.
It was a very interesting finding because usually in other species that we looked at, like humans or bulls or dogs, a frequency of abnormal spermatozoa is higher than 20% is almost invariably associated with infertility. So this was our first clue that the breeding problems that the cheetahs were suffering in captivity had a physiological basis.
Frozen samples of blood from the De Wild Reserve were returned to America for testing.
The next step in our analysis was to estimate the extent and character of genetic variation which was present in the population of cheetahs that were free-ranging and captive in South Africa. And we did this using a technique which essentially has been around for about 20 years, which is to take blood cells and separate them into different components, serum, white cells, and erythrocytes, or red cells, and then to extract from those blood components proteins that can be visualized on electrophoretic gels.
The pattern of the cheetah's genes is clearly seen in the gel photograph. No obvious variation between proteins in 20 different animals when viewed side by side. Whereas for domestic cats, the variation, indicated by the presence or absence of horizontal bands, is quite obvious.
There was no genetic variation in any of the sampled loci whatsoever. Rather, they were monomorphic and virtually identical. They all looked like identical twins, essentially. Or a better way of better analogy is that they looked like deliberately inbred mice.
What was it in their evolution which has made the cheetahs so identical at the molecular level? To find out, the skulls of cheetahs from museums all over America were brought to Steve O'Brien's laboratory in Maryland for detailed measurements to be made. His team of researchers were looking for morphological variation. Many of the skulls collected by Theodore Roosevelt and others in East Africa revealed that throughout this century, the cheetah has had an impoverished gene pool.
There are standard measurements of certain characters, like the length of the skull and the width of this tooth and the width of certain foramina and so forth. And we actually measured bilateral traits, which is the same trait was measured on the left side and the right side, and the difference between those two traits consists of the value which we use to compute the asymmetry of skulls.
This asymmetry in the skulls indicated that the cheetah was in danger of extinction. The cheetah's genes were so nearly identical that there was no guarantee that the many breeding programs would be enough to save them. In a wildlife park in Oregon, the viral disease feline infectious peritonitis, or FIP, swept through the cheetah colony. Although seldom fatal to other cats, for the cheetah there was an epizootic.
Almost 50% of the animals died, and that was clearly the worst epizootic for FIP ever reported in any species. And we were devastated as we watched this, but we were also rather convinced that the genetics that we had been observing up until this point had something to do with the sensitivity of the population. What we think happened is that the virus simply got into one cheetah, learned how to abrogate or get around its immune defenses or whatever it has, and then it looked around the park and they were all the same, and it just went through the park like a train through a station. The loss of genetic variation probably did take place about 10,000 years ago when many species went extinct. And at that time, possibly one or two pregnant females survived and maybe even had several bottlenecks took place that led to the modern population, which now consists of somewhere around 10 to 20,000 animals. Then we also think, because of the differences we saw between East African and South African animals, that there was a second founder effect, possibly as a result of overhunting in the South African animals. And that's the reason we lost the few variants that were present in the East African population. But we think that the major burst led to a population that looked like the East African subspecies. I think that probably the cheetah has a modest prognosis.
Let's have a look at these snails from Hertfordshire.
At the University of Nottingham, Brian Clark has made a second home for his favourite snails. The genus Partula inhabit the Pacific island of Maria. Brian's experiments on their molecules will measure their rates of evolution.
But no sign of young.
Here in November, so we're comfortable.
Being an island population, they've evolved quickly, developing into a diversity of species. And this was first recorded by the American biologist Henry Crampton in 1932. His monograph was the starting point for what's become one of the most comprehensive studies of evolution. The central Pacific island of Maria became the main site for the fieldwork, which began 25 years ago. The genus Partula lives all over the island, and over 1,000 sites were sampled. One of the last sites to be visited was in the southwest corner of the island. Throughout the project, Brian Clark has worked with two colleagues, Jim Murray from the University of Virginia and Michael Johnson from the University of Perth in Australia. But for this visit, he's collecting alone. Brian Clark.
Originally, we went to Maria to study the genetic variation within the species of snails that live there. When we got there, we discovered that what had been described as species were rather uncertain in their position. And so before we could do the studies on the variation within the species, we had to define what the species were. We go and find the kind of habitat in which partula live, which tends to be dense jungle with a lot of undergrowth. And if you after a while, you get to know which places will have snails in and which won't. So we go to the locality and take a random sample of what snails are present, make detailed recordings of the species of plants, the distribution of plants, which plants the snails are sitting on, the aspect of the locality, and as much detail as we can about the habitat, because that will give us clues about which particular habitats are favored by which particular species. When we've done that, it's necessary to locate very exactly where we are. Now, this sometimes is quite difficult when you're in the woods and you've got no reference points and there may be no roads or tracks. So what you do then is go and take bearings on mountains and locate yourself by back bearings. Now we're going to go to another suitable place higher up the valley. There tend to be more species of snail present in locations at higher altitudes than there are at lower altitudes. And particularly you find lots of species in places where there is this climbing pandanus plant, which, as its name suggests, it climbs up other vegetation and it has these rosettes of leaves, and very often in the center of the rosette you will find snails.
So, in the higher altitudes of the island, especially along the steep mountain ridges, there are a number of different snail species living in the same area. For instance, the mountainous region contains 4 separate species: Partula saturalis, Partula tenuata, Partula mirabilis, and Partula toiviana. They mapped the distribution of species over the whole of Maria. Some were found throughout the island, like saturalis and tenuata. Others were confined to certain areas, like mirabilis and toiviana. Discovering their overall distribution was only the first step in their study of evolution. The evenings are for collecting thoughts, a chance to reflect on the fieldwork.
The first things one notices about the snails are their colours. While there is a great diversity of colours within any one species, there are certainly colours that are characteristic of individual species. But there are other obvious characters. One of them is the way the shell coils, whether it coils to the left or coils to the right. We need to know what their relationships are with each other. And that requires that we send the snails back to the laboratory and hope to reconstruct the evolutionary history of the species on the island.
At Nottingham, Brian makes measurements of the snails and studies their internal anatomy. He sets up crosses to see if groups interbreed and so belong to the same species. Gel electrophoresis measures the degree of molecular variation within species and between species. Up to 20 different proteins provide the basis for comparison. These laboratory studies are combined with the field evidence. But early on in the work, Brian Clark capitalized on the simplest of his field observations.
The best predictor of what a snail will be like is what other species of partula are there. And perhaps the best example of this is the coil of the shell. Some of them coil to the right and some of them coil to the left. And you can actually show that one is right-handed or one is left-handed. And they're not just left and right coils, their whole bodies are mirror images of each other, so that the right-handed form has its genital aperture on the right and the left-handed form has its genital aperture on the left. And when a left-hander and a right-hander try to mate with each other, they get into difficulties because they get tangled up, whereas a right can mate happily with a right and a left can mate happily with a left. And we know this is an inherited character. We've done the genetics. And if you look at the patterns of right-handedness and left-handedness on the island, they make sense.
Partula saturalis, which is widespread on the island, is found in right and left-handed forms, but only in distinct areas. Left-handed snails are almost all in the north of the island, and right-handed ones are in the south. This geographic separation is an important clue to the relationship with other species. In the south, Saturalis lives in the company of left-handed snails, such as Partula moriana and Partula toeviana. So here are a number of species, all living within the same area, but evolving separately.
It appears then that snails go right-handed when they're in the presence of another form of partula which is left-handed. In other words, what we observe there is, if you like, frozen in the moment of time, the evolution of an isolating mechanism which reduces the chances of the two forms mating with each other.
These two twists for partula offer a rare glimpse of how evolution takes place. The deduction that the partula species found on Maria have evolved there was confirmed by a re-examination of Crampton's monograph. He classified the snails as D or S, representing the Latin for right or left-handed. Sure enough, at sites where two species were found, their shells were oppositely coiled. So, partula species have evolved where they're now found, on the island of Maria. The variation in their molecules will be a measure of how they're related. As Maria has neighbouring islands, a molecular comparison of the separate island populations was undertaken. The similarities revealed by enzyme analysis produced groups of snails, as if they formed a family, distinct but related. This is the analysis for the Moraean species. But what about partula living on neighbouring islands? After studying the Marian populations, a similar analysis showed a closely related group inhabiting Tahiti, just 15 miles away. This clustering by geography is what you'd expect from groups of species evolving separately on these two islands. The molecular data suggests that the individuals on Tahiti are more closely related to each other than any of them are related to the snails on Maria. So the combined field and laboratory evidence has unravelled the wider story. Within the Tahitian chain, the evolution that has taken place was confined to its own island. Next, they worked out the rates of evolution. When the Japanese geneticist Motoo Kimura first calculated the rate of base changes in the DNA coding for hemoglobin, the result was a surprise, a high rate of one base change every 2 years. This rapid change in the DNA triplets led to his neutral theory, a model for evolution at the molecular level. Compared to the way we perceive the flow of time, molecular evolution is very slow indeed. But for many proteins, this rate, a base change every 2 years, is constant. There's a molecular clockwork in nature. The Society Islands are all inhabited by partula, so the molecular clockwork for each island population could be worked out. It was from these older islands of the Society group, like Bora Bora and Raiatea, that species migrated southeast to colonize the emerging landmass of Maria. Geologists worked out how old the islands are by dating the rocks. Maria surfaced from the ocean 500,000 years earlier than its neighbour Tahiti. This progression, the geological history of the islands, provides a maximum timescale for species on each island to have evolved. But there's another record of each island's history provided by its species. The molecular variation revealed by enzyme analysis showed that changes have accumulated at a different rate for each island. By comparing them over a period of 1 million years, the rate of evolution is roughly constant. But it seems to slow down for the older island groups where there's been a longer time available for evolution. So for Partula, the molecular clock runs at slightly different rates from island to island.
So we have an imperfect clock. If you believe in the neutral theory of evolution, then it's very easy to explain this clock. But if you believe in the selective theory, it's something of a challenge. How is it that when organisms are subjected to all kinds of different selective forces, yet down at the gene level, the clock ticks away? And if we make models in the computer, subject them to what seem to be realistic selection, we find that at the gene level, evolution is much more regular than it is at the level of the organism.
This could happen if single molecule changes in the genes were to have disproportionately large or small effects on the whole organism. This loose coupling would allow molecular changes to proceed at a regular rate under natural selection.
They are, the bigger they are.
That's it,
And
yes.
the size of the box in...
Brian Clark is developing more sophisticated models of natural populations to draw links between selection at these different levels, from morphology down to the gene.
And Urufara valleys in particular.
It's a different perspective from the neutral theory, which draws attention to molecular changes and the random patterns of evolution that they produce.
In trying to explain evolution, we're faced with two competing theories. One of them is the neutral theory and the other is the selective theory. The challenge now is to design experiments which will discriminate between them. The problem is that if you believe in the neutral theory, you are not disposed to do experiments because you believe that everything is indeed neutral and you won't find an effect. If, however, you believe in the selective theory, you will want to go out and discover the selective agents and work out the mechanisms of their actions.